Hi, everybody, and welcome to the Plant Physiology webinar on how respiratory energy can drive everything. Today's webinar is a panel discussion hosted by Steven Burgess and featuring Ulshan Bata, Wolfgang Busch, Keira Havens, Brian Leong, Charlie Messina, and Kristen Van Gelder. We've organized this webinar to recognize the plant physiology focus issue on respiration, and respiration is the topic of today's webinar. This focus issue is the April 2023 issue of Plant Physiology, which was edited by Andrew Hansen, Zoran Nikolsky, Henry Miller, and Danielle Way, and it's online now. We will post the recording of this webinar to YouTube, and if you visit the Plante YouTube channel, you'll find lots of other recordings of Plant Cell, Plant Phys, and other webinars we've made. We'd like to make a special thank you to the members of the American Society of Plant Biologists whose membership dues support this series. If you're interested in joining, we have a code, PRESENTS10, that will give you 10% off your membership dues, and we really appreciate the members and their support. If you have any technical problems, try checking your internet speed or dialing in, and please put your questions for the speakers into the questions box, and we will be reading those aloud. And now I'm going to hand this over to Steven.
Thanks, Mary. Thanks for joining us, everyone. My name is Steven Burgess. I'm really excited to be moderating today's webinar. As Mary mentioned, we're celebrating the plant physiology focus issue on respiration. So this is a really important topic for a number of reasons. Firstly, respiration is central to plant metabolism, and it consumes roughly half the carbon fixed by photosynthesis. This is particularly relevant to plant engineering approaches, as those that require carbon and energy, but also in terms of crop adaptation to climate change, because when the climate warms, rates of respiration will increase, which has implications for plant performance. So again, as Mary mentioned today we have a panel of experts, and we'll be delving into this idea of energy budgeting. Okay, so we'll explore how you can use something called a Fermi calculation to get rough, robust estimates of potential success of engineering projects. We'll also discuss the importance of communication between disciplines and the need to build connections in order to tackle grand challenges. If we get time, we maybe also go into thinking about how we can potentially standardize metrics of success between different individual disciplines, because these do vary. So the format for today is a panel. We'll start off with the panelists introducing themselves and their work in the area. Then we'll dig a bit deeper into two grand challenges, hopefully, including carbon sequestration and nitrogen fixation, before going into a bit more detail about energy budgets and how this relates to respiration. Once we've done that, we'll open the floor to questions. So again, please feel free to enter those into the Q&A, and we're trying to get to as many as we can throughout the hour. Okay. So with that, I'll hand over to our panelists, and if they want to introduce themselves, then we'll go from there. So I think it's Brian, if you want to start.
Sure. Hi, so my name is Brian Leong. I'm a postdoc at the University of Florida with some of my colleagues working in Andrew Hansen's lab. So we are primarily focused on using continuous directed evolution systems to try to engineer plant enzymes. And so Andrew has a number of different hobbies, and so a lot of calculations are one of them. So we'll tell you a little bit about that.
Yeah, hi everyone. My name is Kristen Van Gelder. I'm also a postdoc in Dr. Andrew Hansen's lab here at the University of Florida. I'm originally though from Canada. It's where I did my PhD. And like Brian mentioned, we work primarily on continuous directed evolution, and I'm specifically working with some thiamine biosynthetic enzymes from microbes.
Hey everyone, my name is Ulshan Bata. I'm originally from Germany. I'm also a postdoc in Andrew Hansen's lab at the Department of Horticultural Sciences. And I'm interested in metabolic engineering and continuous directed evolution. I mainly work with plant enzymes, short-lived plant enzymes, and apply continuous directed evolution to make them live longer in order to cut respiration in plants.
Right, maybe I just continue. So my name is Wolfgang Busch. I'm a professor at the Salk Institute for Biological Studies in La Jolla, California. And I've been working for many years now on the genetic and molecular basis that underpin root growth and the responses of roots to the environment. And I'm also the executive director of the Harnessing Plants Initiative at the Salk Institute, where together with other Salk plant biologists, Joe and Corey, Joe Noel, Julie Lott, Michael and Joe Ecker, we're really trying to leverage our science program to understand how we can make plants better to store more carbon for longer in the soil and to sequester significant amounts of carbon to mitigate the climate crisis. And so we do this, and I'm going to talk about this a little later in a number of different ways, and I'm looking forward to the discussion.
Last, I'm Charlie Messina. I'm a professor with the University of Florida, horticultural department. I come from a kind of a different end of the spectrum. My focus is on predictive breeding. So I look at the whole plant physiology in a way to understand adaptive traits. And of course, respiration it could be key, as it was a little before, to increase productivity or mitigate part of the problems with climate change. In the past, I worked with corn, and a big question is, as you might see in the recent paper that Andrew published with some of us as co-authors, the radiation efficiency has been going up, and we can explain everything from photosynthesis. A big hypothesis is what was the role of respiration in there.
Great, thank you very much, all. With that, I think Wolfgang, if you want to maybe go into a bit more detail about your project, and then we can move on from there.
Absolutely. So let me share my screen. Did that work? Do you see the full screen?
Yeah, that's perfect. Okay,
excellent. So I'm going to talk a little bit about the Salk Harnessing Plants Initiative, and because this will be relevant for the discussion when it comes to the numbers and what we might expect from plant-based carbon sequestration. And we basically came together a couple of years ago. At the time when I joined the Salk Institute, all of us plant biologists here, and thought, climate change is the worst thing and the most pressing challenge that our world, is facing. And we all know, of course, being plant biologists, that plants have numerous connections to this, starting from carbon capture, but also for production for food, feed, and fiber, and adaptation to the climate. And so we all came together and started thinking about what can we do? How can we leverage the science that we do to potentially address this? And so this is kind of what it boils down to, why it's so important to think about climate change and plant biology. And this is embodied in this chart from the IPCC 6 report assessment that actually shows us what will happen in terms of the average temperature increase on this planet when we will continue to emit the way we do now, which is like this red line. So we are on track for 3.2 degrees Celsius increase of average temperatures, which is tremendous, and will have a number of very negative consequences. Or what will be needed in terms of changing the CO2 or greenhouse gas concentration in the atmosphere to achieve 1.5 degrees Celsius or 2 degrees Celsius global warming by the end of the century. And as you can see, those two curves of modeled pathways towards this scenario, they all lead through a gigantic reduction of the greenhouse gas emissions. You see that on the y-axis, that in order to get to the 1.5 goal, we need to go from 55 gigatons of CO2 equivalent emission per year to around 12 by the mid of the century, and that is enormous. So So the question is, what can we do to prepare us, and what can we do to mitigate this? And so we kind of agreed, like one important pathway that everyone needs to think about is the climate adaptation of crops, because the temperatures will rise. There's no question about it, whatever we do. But also agriculture, and that being the major, and human endeavor with plants on this planet, we need to think about agriculture emissions, avoidance, that is, and nitrous oxide, for instance, and relates, I think, to some of the points we'll discuss with nitrogen fixation, where potentially some agricultural N2O emissions can be avoided. And the third one is plant-based carbon sequestration, which we, at the Salk in our initiative focus on, because in order to go from 50-something gigatons per year to 10-something gigatons per year, it is not imaginable that actually the energy transition will be fast enough to do that. So the question is, how can we actually draw down additional carbon dioxide? And plants, as we all know, are the world, champions of catching carbon dioxide from the atmosphere, and they're already there. And we culture them on, at an enormous scale, on hundreds, or on billions of hectares worldwide. Okay, I'm going to skip that. Just showing you that if you actually take, and that is something that convinced us that we should actually get working on this. If you think about the human emissions with 37 gigatons of CO2 per year, and you think about the capacity of nature to actually absorb CO2, the human emissions and the net human emissions that are not absorbed by nature already actually are dwarfed by the capacity of natural systems to take up CO2. And a significant fraction of this natural ability is constituted by photosynthetic organisms, mainly plants. Another factor why it's important to think about crops in particular, because those are the most widespread, species that humans cultivate, is something else. So if we think about the fact that plants are very good at capturing carbon and putting it into biomolecules and biopolymers, the question is, how can we actually make it stick somewhere? And the somewhere is in the soil. And then the next question comes, might there actually be capacity in the soils to store the enormous amounts of carbon that need to be stored if we want to draw down carbon dioxide to have a global impact? And this study here kind of convinced us again early on that there is actually a lot of merit to think about this, on how to store carbon in the soil that has been captured by plants, and think about ways to make it last for longer. And that is a study in which they used actually historical data and modeling, again, another theme of our panel discussion today, to kind of figure out how much carbon that had been in the soil. The soil is a huge carbon pool holding 2 to 3 times more carbon than the entire atmosphere, already has been lost because we, as human and historic humanity, have converted natural lands, for instance, into agricultural lands, and in this process released already a lot of carbon. Think about the prairie lands that are now our corn and soybean belt in the Midwest. And so this modeling exercise from 2017 from Jonathan Sanderman et al. Actually showed us that actually agricultural soils, that's not only croplands but also grazing lands, have already lost 133 gigatons of carbon, which constitutes 17% of the atmospheric carbon pool. So that is, of course, bad because we have to release it, but it also tells us that there is actually enormous capacity of agricultural soils to store carbon. So one idea would be, if we just could backfill this, that would be enormous. That would actually be a tremendous part of what we need to do to limit global warming. Okay, so we came up based on these, on the reading of these studies, and also running our own kind of Fermi calculations. We can talk about this later on, like the back of the envelope calculation, with some ways of how we thought we can stabilize captured carbon that's captured by plants, using a couple of traits. And those were only like the traits that we came up with, I think there are many more opportunities to think about. The first thing about this is we focus on the root, because root-derived carbon is basically much more likely to stay for longer in the soil than shoot-derived carbon or litter. There are studies that have looked at the lifetime of shoot and root-derived carbon, a number of this. So we focus on the root, and we're focusing on 3 different traits, which are root mass, the amount of carbon that is in the soil. If root carbon sticks around for longer, more carbon in the roots will help to trap more carbon in the soil. But then importantly, the 2 traits or 2 characteristics that relate to the permanence of the carbon in the soil. So for instance, in the upper soil layers that are tilled, or even in no-till scenarios, they get turned over much faster. So the average lifetime of carbon in the top 30 centimeters is maybe less than a year to a couple of years. But if you go below 30 centimeters, the average lifetime of carbon gets much, much older. So you can get down to hundreds of years or thousands of years. So that is why a second trait that we focus on is the placement of roots in the soil, the root depth. And a third one relates to the biochemical composition of roots, and that is how this connects so well to the study that we are talking about, or the paper today. That is suberin. Suberin is a natural formed biopolymer that is formed in specific root tissues. There are studies that have demonstrated that it stays around for much longer. The question why is still debated. Is it its own biochemical recalcitrance? It's very oxygen poor. It's very long chain, fatty acids, and so it's hard to break up, in particular under low oxygen environments. But there might be other properties, such as chemical interaction with soil minerals and preferential, aggregation into soil aggregates that might help it to live longer. So nevertheless, for the reason why we focused on trying to understand how we make more suberin in the root by engineering tissue types and the biochemical and basically the biochemical pathways that plants use to build suberin. And so those are our main traits, and we have built over the last couple of years a research program that spans from molecules to whole organisms, and from the lab to the field. We work on model plans for understanding these traits better, fast prototyping, and we work in crop plans where we genetically engineer but also harness the diversity using massive GWAS approaches for these traits. And so we are moving full steam ahead and have identified already more than 30 genes for these different traits that can enhance this. And so the next step is to really test this. And another next step, and something that we are really interested in, is into using more modeling, in which cases that might be really beneficial for crop species to implement this, and might be a win-win for farmers and the environment. All right.
Thanks, Wolfgang. That was great. This is really fascinating. So we'll come back to that pretty soon. Keira, do you want to briefly introduce yourself and then maybe give you an introduction to what you guys are working on too?
Yes, absolutely. And I'm so sorry for the delay in joining. I'm traveling and just took me a little bit to get in. So appreciate being here. Really just excited to see this work being discussed more broadly. My name is Keira Havens. I am with Pivot Bio. I am the senior sustainability project manager for the group. And what I do is I look at the impact of some of these of our microbe, which takes nitrogen in from the air and produces ammonia at the root of the plant. And so that plant is then able to take that ammonia up, and growers love this because their nitrogen doesn't get lost in the crop growth. And the earth loves it because, again, that ammonia goes into the crop instead of into the water and air as it gets lost. So that symbiotic relationship of a free living diazotrope on the root surface of a plant root, that's a very interesting aspect of the carbon discussion, right? Because those microbes feed on carbon, and that's what fuels the nitrogenase and the nitrogen fixation. So pleased to be here just to talk a little bit more about the importance of this research and what it means to kind of look at that energy budget as a whole for the future of the tech.
Okay, so moving on, Brian, do you want to maybe talk a little? I think you were first, right, in terms of your team talking about things. Or
I think Olshan.
oh, it's Olshan. Oh, okay,
Yeah.
so.
Yeah, so I hope you can see the screen. Okay, perfect.
Yeah.
So, as most of you may know, that plant growth can be simplified as the conversion of carbohydrates, nitrogen, sulfur, phosphorus, and other nutrients to new biomass. And of course, these processes require energy. This holds also true for many synbio interventions in crop plants, for example, for the production of compounds for protein expression, for nitrogen fixation. And this energy comes from respiratory oxidation, also known as dark respiration. I will not go too much into detail about the involved pathway. I just want to point out that glycolysis, the pentose phosphate pathway, the TCA cycle, and oxidative phosphorylation, they all together produce ATP, NADPH, and FADH, which together make 28 ATP equivalents per invested hexose. So this is the energy a plant can deal with, that can use for downstream biosynthetic pathways, or for any pathway in the plant. So I also want to mention that the numbers we applied in the downstream calculations of our paper are really unconventional numbers, but led us to inescapable conclusions. So would like to mention that plants during evolution evolved under nutrition limiting conditions, in particular nitrogen limiting conditions. So there was a surplus of carbon. But since the Green Revolution, and as we apply sufficient amount of fertilizers, including nitrogen, phosphorus, and potassium, plants have enough nitrogen, and they're no longer nitrogen limited, but rather carbon limited. So the balance between the elements kind of changed. So now I want to talk a little bit about the processes, and what processes are fueled by the carbon budget the plant has available. So 50% of the carbon budget in a plant goes to biomass production. Roughly half of that is bioharvestable. And the other half of the carbon budget goes into respiration. 50% of that goes to gross respiration, and the other half goes to maintenance respiration. And maintenance respiration means maintaining ion gradients, for example, but it also means protein turnover, which is breakdown and resynthesis of proteins and enzymes upon mainly self-inactivation, because many enzymes perform dangerous chemistry. They have reactive substrates and products, so they can be damaged. Some enzymes get damaged more often than others, so those are the ones particularly which are costly to the plants. And the entire process of protein turnover makes roughly 10 to 50% of a plant's total energy budget, which means that it's also a profitable target for synbio interventions. And for example, when you engineer enzymes and proteins to live longer so they don't get inactivated that quickly, you may be able to cut respiration, and then the saved energy could be channeled into production of new biomass. So within the paper, in one of the chapters, we asked what could be the biomass gain when enzymes get engineered to live longer, and thereby respiration is cut. So for that, we calculated the ATP cost of turnover for specific enzymes. And for that, one would need to know the turnover rate, which we typically get from empirical proteomics data, and the turnover cost. And the turnover cost, to calculate that, you would use numbers from the classical literature. This number is typically around 6 ATP per residue. So this would be the cost for degradation of an enzyme. This is typically around 1.3 ATP to break down a damaged enzyme, for example, into its single pieces, so the single amino acids. And 5 ATP would go into the resynthesis, so the reassemble of these single amino acids into a functional protein. So together they make around 6 ATP per residue that such a turnover cost. So given a certain number of residues and enzymes that you're interested in, you can then calculate the turnover cost. And then within our paper, we did this for 15 high-cost enzymes and applied a case in which these 15 high-cost enzymes would be engineered to, let's say, live 10 times longer. So it means that the reduction in turnover rate would be 90%. We applied this to our data and came up with a lower maintenance respiration rate, 6.5%. And then you can use these data to feed into crop modeling software. And this was done by our wonderful collaborator, Jeff Amther, who then calculated that that these 15 high-cost enzymes, when their turnover rate would be reduced by 90%, that a model crop could produce 3% more grain carbon, again, only when we would engineer 15 enzymes. And I think this is quite interesting. This is a huge potential to cut respiration in crops and to save energy. And what the plants can do with such saved energy, this was explored in more detail by Kristen and Brian.
So I'll kind of take you through the next part on biological nitrogen fixation. So Olshan shared with you guys where all the ATP and the reductants come from and how we calculated, the ATP costs for hexose. And what can you do when you can, reduce protein turnover and gain this extra carbon in your budget. And so one of the areas that we looked in is to biological nitrogen fixation. So first we had to start off calculating what the actual cost is and going through the literature and figuring out, what's the reductant and the ATP cost for that, and then converting it into a hexose cost. So we did that for ammonium and nitrate, which can be found freely in the soil. These are obviously your lowest costs. So we put into calculating this the cost of transporting it and uptaking it, and of course for nitrate, the reduction, because nitrate can't be freely used as is. And then we also calculated for more bioengineering strategies. So looking at implanted nitrogenases, what would be the cost if you could put a nitrogenase into the leaf, so in the leaf of a maize, and what would be the cost for that? And we calculated them for kind of two different scenarios, one where we've got a lower cost in terms of ATP, and one where we've got a much higher cost in terms of ATP. And so what would be the two costs between those? And then our last calculation, we went into looking at the diisotroph associations and what's the cost for that, for converting nitrogen gas. And so again, we calculated at these low range and high range of ATP in order to do the conversion, the uptake. And so from these costs of biological nitrogen fixation, what we really wanted to know is if we can replace nitrogen fertilizers, so nitrogen fertilizers that come from the Haber-Bosch process, what would be the yield penalty that would impose? Again, we're using our maize as our model crop here, but there was a lot of other assumptions and things that we had to figure out as well before we could go into calculating what it would cost to completely replace nitrogen fertilizers. And so some of the kind of the assumptions and that we made before we did these calculations is that, so the maize grain yield is about 11 tons. And then we're looking at, all of the nitrogen content and what application of nitrogen is done per hectare. and how much of that can actually be retained, which is not all of it. So it's about 120 kilograms of nitrogen per hectare is retained. And so that would then, of course, we would want to completely replace with some sort of BNF. And so, again, we kind of went through the cost of the yield penalty. So what's your grain yield when we've got about 1.4 grams of hexose per gram of maize grain? And basically, again, we walk through those calculations, looking at low and high ATP scenarios. What would the yield penalty be for bioengineering and nitrogenase into the leaf system? And the yield penalty is quite small. If you look at using nitrate as our basis, you're getting about that 11 tons per hectare. There's not much of a decrease in terms of engineering nitrogenase. Where we see the greater yield penalties start to be imposed. So when we look at engineering the better diazotroph associations, again, looking at that low and high ATP cost. But we also looked at two scenarios for diazotrophs. We know that they're supplemented with photosynthate from the roots that's released into the soil that they can then use to fix nitrogen. And so we assume that this photosynthate would be hexose just to make the calculation easier. And so if the diazotrophs could use 100% of that exudate coming from the roots to fix nitrogen, and then you're replacing completely chemical fertilizers with these diazotrophs, what would be the yield that you get? And there isn't much of a yield penalty imposed when you are able to use 100% of that photosynthate. Of course, that's not very realistic. We know that they don't use all of it. They use just a very small portion of it. And so a bit more realistic, but maybe still straying on the optimistic side, is if the diazotrophs use just 25% of that root exudate for nitrogen fixing, what would be the yield that you get by completely replacing the chemical fertilizer. And at the highest ATP cost for this, you're looking at almost an 80% reduction in yield. And of course, the yellow bars here, we just calculated what would be the cost to actually produce the exudate as well for the complete replacement of the nitrogen fertilizer with these BNF strategies.
Okay. So in order to wrap things up, my section was primarily focused on the carbon capture and sequestration compounds. So this is a technology, as Dr. Busch mentioned, that's been gotten a lot of play in terms of discussing how to try to mitigate climate change. Plants have been proposed, as he mentioned, as one of the ways to try to do this via the production of recalcitrant compounds like sporopollenin, that's often found in pollen, or suberin, that's found in plant roots. And plant roots are of one area of interest because of the ability to sequester that carbon in the roots and therefore in the soil, and try to sort of increase or stabilize the carbon storage for a longer period of time. Now, our interest was trying to understand some of the calculations behind this and what the potential is here using the current technologies. So the question is, how much carbon can you actually sequester in plant roots? And what do some of the economics look like? So we really do need to kind of get society and farmers on board with these approaches, because in principle, if they're not buying in, it doesn't matter if we can produce the plants that can do it or not. So the first question was sort of, what are the energy requirements for making some of these compounds? And so we started by coming up with an estimate for sort of what the average monomer of these two different compounds would look like. So you can see on the left, sporopollenin and suberin. And these are sort of rough estimates or average structures of these compounds. And so at that point, you can then sort of go back to the metabolic map that Olshan showed before. And starting from hexose, you can then try to figure out how these compounds could be synthesized. So the first thing that we did is we looked at the structure of the compounds and tried to break it up into its component parts. So many of these compounds are lipophilic, so they could be derived from things like lipid biosynthesis or from polyketide synthesis. Some of them are almost certainly derived from like phenylpropanoid biosynthesis. And so we can start from there, starting from hexose, we can work our way down through these biosynthetic pathways, and using metabolic maps from like KEGG or MetaSyc, Psych, you can start to get an estimate for the energy cost. Okay, so that's a lot of what I did for this paper. So once we got the estimates for all of the component parts, and you can see here, we had estimates for ATP, reducing equivalents, and then hexoses for both the energy as well as the carbon backbone calculations. We get an idea for what that monomer costs. Okay. And then based on the average or the molecular weight of our monomers, we can then scale up and say, well, how much does it cost to produce a gram of these molecules? Okay. And so based on that, the proposed monomers and the molecular weight, we can then determine sort of what what it would cost the plant in terms of energy. So you can see a plot on the right, which is an estimation of the hexose cost for different types of biomass production. You can see stover biomass on the left here at the low end, which is increasing as you move to other types of biomass, like producing just protein or triacylglycerol. And what we found is that sporopollenin and suberin were on the very high end in terms of the amount of hexoses necessary to produce a gram of this material. Okay. Knowing these ratios allows us to do a couple different things. One of which it allows us to determine what the cost of the plant is. And so as some of my colleagues have mentioned, we've used maize as our model. And so we estimated that approximately, based on the literature, with some engineering, you could get probably reasonably 10% of the root biomass as these CCS polymers. Okay. And then digging into data from the USDA and previous studies, we were able to look at, determine the maize acreage across the United States and say, okay, well, what is the average root biomass in a given area? And then from there, you can estimate how much root biomass is produced. And then saying 10% of that will be our carbon capture compound. We know how much of that is, and then it's just scaling it down to carbon, how much is actually captured. Based on those calculations, we estimated that planting these plants across the maize acreage in the United States, you'd be able to sequester about 0.3% of the total US fossil CO2 production every year. Okay. The other thing that we're really interested in was trying to look at some of the economics, because we're going to have to get farmers on board if these approaches are going to work. Okay. So on the right, you can see a plot of basically two different scenarios. One is a zero compensation case, in which essentially you have to pay for that increased biomass. So if you're producing carbon capture compounds, it has to come from somewhere. It's not magic in principle. And so if, for example, we were able to use basically the amount of hexose cost for each of these different types of biomass to give us sort of an exchange rate. Okay. So for example, if we're going to take it out of grain income and put it into some of these carbon capture compounds, we can estimate sort of what the grain loss would be, and then what the price would be at different carbon offset prices. And so the zero compensation case, as I mentioned, is sort of paying entirely from other biomass, whereas the 100% compensation case is effectively where if you could increase photosynthetic output or decrease respiration costs, you can try to channel some of that energy into it. And so effectively, these are sort of like you're completely paying for it from other like photosynthetic gains, etc. So there were sort of two major findings. We found is we lost about $64 per hectare approximately if you're taking it directly from the grain income in the 0% compensation. Okay, so at that point, you can get an idea for what the carbon offset prices would need to be in order to try to mitigate some of that loss of grain income. We think that's sort of the worst case scenario. It almost certainly wouldn't be that severe, but it does sort of give you a floor. Okay. But what that means is that the carbon offset prices in that case would need to be close to $200 per ton to offset just the loss in grain in the worst case. Now, the upshot is that in the 100% compensation case, if you can start to derive some of that energy from other sources, it provides a nice subsidy, especially as the price of the carbon offset increases. It provides a nice subsidy and incentive for farmers to grow some of these crops. And so we think that that can also help stabilize some of the fluctuations in prices for crops and things. Yeah, so.
Thanks. This is great. So quickly, Charlie, if you maybe could provide, as your background is very different, it'd be great to get the perspective of agronomists as well on the idea of carbon sequestration and energy budgets.
Yeah, the models we use essentially use the maintenance and growth respiration, so we can do this analysis and as we shift growing seasons. So one of the projects we are working with a number of universities, and Wolfgang is part of it, is this CIRCA project, trying to reimagine how we should grow corn in order to reduce respiration, because we can grow it in colder environments as temperature increases, but also how we can change the growth respiration, for example, by modifying the protein content in the kernels. That's been happening for 100 years of selection. Protein has been going down. Therefore, you saw the calculations, I think it was Christine show how much it cost. So it's an expensive polymer to synthesize. So if we reduce the protein concentration, focus on the energy, then part of that could be subsidized to produce compounds we want to sequester, or it could go to yield. But essentially, that's a component that is easier to manipulate. The other one would be really interesting, and we can do this analysis, is on matching a little bit better the supply and demand of nitrogen. For example, there's a lot of uptake of nitrogen early in the season that has to be stored, and it has to be remobilized. And you saw how much is the cost of just every of these reactions have a cost. So the mathematical models we use in prediction, in genomic prediction, in plant breeding, they're based on this calculation. So we can think about what are the crop designs we want to use. And as the SynBio provide information about the opportunities to manipulate the pathways, we can update this information in the crop models as we think about the different possibilities. I think you mentioned also in the chat about no-till. That's a little bit of a tricky question. In many parts of the United States, you talk to the farmer and say, that's wonderful, but if I do no-till, I cannot plant because the soils get too cold. So that's a little bit more difficult question to answer. Certainly, no-till is using less carbon and contribute to reduce the carbon emissions. But hopefully that answer or give a little bit of perspective of what you ask.
Oh no, that's fantastic. So yeah, we have questions in the Q&A, and just quickly before we get to that, I think an important point is that so the model, as was mentioned, is based on the assumption that crops are carbon limited. This is true in most instances, but I wonder if some of the panelists may be able to comment on the potential implications of this, and if other upsides or things that should be considered. Yeah, Wolfgang.
Yeah, I mean, first let me say, I think it's really fabulous, in particular for the early career scientists, to get at this like huge and tremendous question, and also run the numbers, which is often I think very scary because they are so clear. And so I think one thing that I want to say is this is so important, right? And because modeling, is based on on a set of assumptions. Every model is. And our processes, right? There might be, in particular, in early days of modeling where less is known, you might get like a kind of like a broad sense given a set of assumptions, but over time that that might change. And given other models, I think in the soil science community, for instance, the carbon sequestration potential through plants, have been quantified. It's very much at the other end of the spectrum. I think the more modeling happens, the better we'll be able to converge and also identify the opportunities. And this is what I want to mention. I think it is true that in my view that plants in some circumstances are carbon limited, but often in particular, maybe not so much nitrogen, but water might be really limiting in many circumstances. And just like an empirical point of data I want to throw in is the yield potential, right? The national corn yield champions, they basically can get up to 500 bushels per acre, but the national average is 170 bushels per acre. They all see the same amount of carbon dioxide, and they also, in many instances, have very have the same light interception perhaps. And so I think something to consider is that there are actually a number of factors that might be affected by roots, such as the ability to gain more water through deeper roots or withstand drought. Suberin is actually used naturally by plants to increase their drought resilience, and is used in many plants in either very wet or very dry environments. So I think that is important to consider. And so I believe there is actually an opportunity that you can get a win-win when you think about the root system that is better to withstand stress and can take up more water and maybe some of the other nutrients that might be limiting, you might actually have more of a carbon budget, because the roots part will enable the shoot part to basically capture more carbon. And so why that is really a very difficult exercise to model, these types of model will actually allow us to say, oh, maybe we should focus on this crop species first, where the gains, the win-win might be better, and in these areas. And I think Charlie has done some really interesting work, that resulted in the Aquamax, thinking about these types of adaptations. So modeling is super important. It's important to do it, and I love what you have done, and it has been done in that paper. But and I think modeling is the key to prioritize and getting to better numbers.
Thanks, Keira.
Yeah, thank you, Wolfgang. I'm so glad you brought up that it's a dynamic system and that it may be possible to increase that carbon budget, right? As you said, we see that when growers have higher yields, it's possible for that much more biomass to be produced, for that much more respiration to occur, for that much more carbon to come out of the air and into the plants and possibly down into the roots with the microbes. And I do just want to say to echo the point about the numbers. I come from academia. I was a wet lab scientist for a very long time, and now I'm on the commercial side. And we do this work ourselves to model for products, and it's really great to have it happen outside of our little circle, right? This is something that keeps us honest, keeps us, when I read the paper and saw, oh, we're in the ballpark, that was a great thing to see. And knowing how much work it takes to collect all these numbers from all different disciplines and bring them together into a usable format and create a single model. I just want to applaud the team here for this work, because it is an intensive endeavor for sure. I am excited for the next iteration, but making an inventory of what we know right now, that is the place to start. So this is a great addition to the body of literature.
Really?
Well, I would like to follow up on the 500 bushels. We tried to replicate that, and we can't. There's some sort of magic carbon fixation there. However, we've seen that this RUE, whatever is, I think it's a combination of canopy architecture, leaf photosynthesis, and respiration and partitioning going on. But we think there's still room to continue increasing that photosynthetic capacity. Of course, there's a limit. I think Dr. Amthor and Loomis did some nice, solid calculations that we can, again, we project what would be that sort of yield potentials. Related to that partitioning and opportunities, I think the dwarfing genes you've seen now in industry, thinking how they can contribute in many ways to manage nitrogen better, but also they are shifting the different sinks in the plant. So some of that could be allocated to the roots, for example, or higher, more costly compounds to think how we can sequester that carbon, right? And finally, I just would take advantage to make a little bit of an advertisement with Wolfgang working on the Circa. If you're interested on the modeling and hopefully we convince you it's important, we'll be looking for postdocs. So that's all.
Right, it's okay, great. We have lots of great questions. We'll try and get to as many as you can. Some of our panelists will start answering also in the chat as we go along. I'll just try and start working through some of these now. So I guess one of the first things perhaps to pick up on is perhaps also you can answer, there was a question about how do you actually go about increasing the lifetime of proteins? How might this work?
Yes. Yeah, we work with the continuous directed evolution system that was developed for yeast. It's called OrthoRep. It's a very powerful tool that applies error-prone replication, translation, and selection in a host system, as I mentioned, in yeast. So we typically use a metabolic knockout mutant, for example, for the enzyme that you're interested in to make it live longer. So you would use a knockout mutant that in particular this biochemical reaction cannot be performed by the platform organism. And then you express your enzyme of interest in that OrthoRep system, and you make the host strain with the metabolic knockout dependent on the performance of the expression of your gene of interest or the enzyme of your interest. So, and when you then select for populations, for cells which can grow faster because they have accumulated beneficial mutations in the gene of interest that allow them to grow faster and to complement the metabolic knockout, you then can select for mutated enzymes that would prolong enzyme life. This is the strategy that we use in the lab. And I think there was a second question, how realistic it is to make those enzymes live 10 times longer. It really depends on the enzymes you're working on. The most extreme example is a suicide enzyme called thiazole synthase that can only perform one reaction before it has to be replaced. So to make that enzyme live 10 times longer, which basically means that it can catalyze 10 reactions before it has to replace, might be a way smaller step than when you start with an enzyme that can catalyze 10,000 reactions and you want to make it to catalyze 100,000 reactions, so 10 times more. So it really depends on the target. But the 90% reduction, so make it 10 times longer, is rather probably on the realistic or more conservative side. In particular, when you work with very short-lived enzymes, so very expensive enzymes to the plants, you would probably rather shoot for making them 100 times live longer or 1,000 times live longer. So I think 90% is, as I said, more on the conservative side. Yeah.
Great. So I'm going to try and combine a few questions here. I think there are a number of great ones with a similar kind of theme. But this is really kind of thinking about what an ideal plant would look like and what the potential is to try and stack different technologies together. So we talked about two things, about conserving energy and then also starting to put this into different applications. I wonder if anyone of the panelists would like to try and address this.
Should I take a first step? So since we are like having this very modest term of the Salk ideal plant, no, but I think stacking would be would be super important. If I imagine all the if I think about all the work that has been done in several institutions about optimizing photorespiration or photosynthesis, if you add up that to a root system that is preferentially leading to more carbon for longer in the soil, that would be really a win. And I also believe, because we all know plants are integrated systems, there might be more than additive effects if you engineer different parts of the plant, right? A certain root architecture in terms of water, for instance, nutrient capture ability, will be able to sustain photosynthetic activity at a certain level. So if you want to really move that up in an environment that at least temporarily water limited, you want to engineer the root and the shoot. So I think, I believe there will be hopefully a community effort to basically integrate a number of the excellent ideas that are getting out there to really do some more integrative engineering. But a first step is always to look at the individual trait and test it in a certain crop species. We also should note, this is here really focused on corn, but if you think about the global distribution of crops, and I always think about this from the carbon sequestration perspective, right? We have like 1.6 billion hectares worldwide that are basically planted with crops on this earth. That is enormous, right? But there are many different species like soybean, corn, sorghum, wheat, and they all come with their different challenges. And I would be very, very surprised if you could use the kind of the increase of certain traits the same way, right? And so to achieve something. So you need actually, and I think modeling again is the way to go, to see what is actually the best bang for the buck in terms of increased yield or keeping yield kind of like neutral, and things like carbon sequestration or nitrogen use efficiency, or feeding microorganisms in the soil to do something great like fixing nitrogen. And I think modeling will play a huge role, because every crop is different. The growth, the season length is different. So there are so many regional parameters. And so modeling, again, coming back to the theme here, will be key to prioritize and to think about which trait concepts are worthwhile to test.
Else want to add anything? Yeah.
I can follow on to that and just say that we need every tool that's out there. It's mentioned in the paper that the paper looks at nitrogen produced by microbes either at 100% or 25% capacity. And the truth is that growers are going to be using both kinds for a long time, until there is enough capacity to switch over entirely and preserve their yield. Because for them, their sustainability directive is to make sure that they are able to continue producing what they grow and maintain their productivity. It's to ensure that their operation is financially sustainable going into the future. So each and every one of these tools is very likely to find a place down the road. There's a ton of space for innovation here, and a ton of space for developing new tools that make the process of growing and producing biomass for purpose. And let's just think about a future where we have transitioned away from petroleum to a biomass focused economy. Just like what was being said a minute ago, right? These tools will have different places for different crops, different locales, different agronomic situations. And I think there's a lot of power in building out a multiplicity of avenues to approach this carbon takedown and sequestration avenue.
Cool. So quickly from Charlie, and then I wish we had 2 hours. We have plenty of questions, and we'd love to talk for a long time. But Charlie, maybe you can.
I think we like to add is also these numbers that Kristen provided. It's also make us think that some of the times the controls are not at that level. We shouldn't make the bacteria fix nitrogen, because that's perhaps too costly. Maybe what we need to do is grow 2 crops in a sequence. So the control of that nitrogen fixation is not at the roots level, but at the system level. So That was quite enlightening anyways. I just want to highlight Christine's contribution to just help us think how we are going to use this set of tools to design systems using all the tools we have at our disposal.
Absolutely. At the system level, at the agronomic level, that's where the impact really happens. I agree.
Hey, I realize we're coming up to time. So I just want to thank all our panelists for a great overview of the topic and discussion. As I said, I would love to keep talking for a long time about this. I think there's a lot of mileage here. I'm sorry if we didn't get to your question. We'll try and answer what we can, maybe potentially through email later on. But thanks again for everyone for participating. And Mary, I don't know if you want to add anything.
I want to say thank you, Steven, and thank you to all the panelists. I mean, I come from the era of molecular biology where we thought one gene would solve all of our problems. And it's really interesting and challenging to hear that was kind of a simplistic point of view we had back then, wasn't it? But really stimulating. And again, thank you to those who put in the questions. These were some of the best questions I've seen in one of our webinars. They were really thoughtful. We look forward to getting back to you. And thank you really very much, panelists. This was very, very stimulating. Bye-bye. Thank you.